Back to my lectures. In today's lecture, we are going to see about structure and functions of plasmid. Now let's see the properties of plasmid in brief. As we know that plasmids are small, double-stranded, supercoiled extrachromosomal DNA present inside the bacterium cell other than the chromosomal DNA. Okay, and that there is an exception of linear plasmids also present in Streptococcus species. Plasmids have ability of self-replication and transmission. It was first introduced by Joshua Lederberg in 1952. It has other names as it also called, we can also call plasmids as replicons, sex factors, conjugates, transfer factors, etc. It have less than 30 genes inside it. It can also act as a vectors in genetic engineering. Its genetic information is not essential for the host, thus can be used as a vector. It can live cell can live in absence of plasmids also. Their presence provide extra beneficial effects to the bacterial cells. It is also known as episome. That means plasmids are also called as episome because they have ability to integrate with chromosomal DNA. We have already seen HFR plasmids and F prime plasmids, like in which plasmids are integrated into the chromosomal DNA, right? So plasmid have one of the property, that is plasmid curing property. That means in cell division, when cells are getting divided during the cell division, plasmid can transfer from one cell to another cell, but sometimes it gets lost. That's why it is called as plasmid curing. That means loss of plasmid is also known as plasmid curing. Plasmid have specific copy number. That means they are present in the cell in the form of copy number. If there is a larger plasmid present inside the cell, that means they will be present in fewer numbers. And if they are smaller in size, that means if smaller plasmid is present in a bacterial cell, then they will be in more numbers. So this is the copied number. And one of the most important property of plasmid we can see is as compatibility. That means two different plasmids can coexist stably in the same bacterial cells. So this is the compatibility of plasmids. So these are the properties of plasmids. Now let's see the structure. This diagram will explain you all about the general structure of plasmid. So this is the basic structure of plasmid, you can see. So first of all, the first structure is origin of replication, or also known as ORIT region. Okay, so this is the region where replication process can start, and it is generally present in AT rich region. That means it has abundance of adenosine and AT nucleotides presence inside the ORIT regions. Okay, because AT bonds can broken down easily, thus helps in the replication process, stand breakage, you can say. So this is the first origin of replication. Then coming to the second, antibiotic resistance gene. That means some plasmids have selectable marker region inside it. So for example, antibiotic resistance you consider, or some chemicals degradative regions are also present in the plasmid. So antibiotic resistance genes are selectable marker regions present inside the plasmid. Some are selectable marker region where some specific selectable markers are present inside the plasmids. For example, you can say lactose metabolizing gene is present in, for example, you can say lactose metabolizing gene is present inside the plasmid. So it is occupied in the selectable marker region. Then the promoter region is present in the plasmid. This is the region where transcription process starts. That means during the process of formation of mRNA, that means from DNA to mRNA, DNA strand broken down and then mRNA will be synthesized by the process of transcription. So this promoter region includes number of nucleotides responsible for the transcription. So transcription will start in the promoter region only. Then the primer side. 5 prime primer sites are present in plasmids also. Then some plasmids have selection region, that means it has specific restriction sites, and some sort of inserted genes are also there, and this is the and their size will vary from 2 kilobase pair to 200 kilobase pair. So this is the region where restriction enzymes will come and bind to the restriction site and will break down the plasmid at this site. And the last is the 3 primer site. So the start of the plasmid will be from the ORIT region and the end will be at the 3 primer site. So this is the basic structure of plasmid present in the bacterial cell. Now let's see the types of the plasmid. Based on the presence and absence of TRA genes, plasmids are classified into two types, conjugative and non-conjugative. And based on the functions, plasmids have different types, that means fertility plasmids, resistance plasmid, colicin plasmid, degradative plasmid and virulence plasmid. Let's see one by one. Depending upon the presence and absence of TRA genes, there are two types of plasmid. First is the conjugative plasmid and second is non-conjugative. Conjugative, that means the plasmid which have the ability to do conjugation. We have seen conjugation in detail, and the link for this conjugation will be provided in the description box. So conjugative plasmids have TRA genes presence inside it, and they have the ability of the conjugation. That means the one plasmid can transfer from one cell to another cell via conjugation process, and the example is F plasmid. Okay, and second one is the non-conjugative plasmid. Here it lacks the presence of TRA genes which are responsible for conjugation. That's why it is named as non-conjugative, because it lacks the TRA genes, and it do conjugation with the help of conjugative plasmids, and the example is col E1 plasmid. Now depending upon the function, first one is the fertility plasmid, or you can say F plasmid or F factor. It has TRA genes, and it has the ability to synthesize the pillars for the conjugation, and it can do self-replication by itself. Then second is the resistance plasmid. Resistance plasmids are also called as R plasmid because it have the genes for the antibiotic resistance, and they are in the larger size and thus present in the low copy number. Okay. Third one is the colicin plasmid. Colicin plasmids are also called as col plasmids because they are also known as bacteriocinogenic plasmid because they produce bacteriocin. Okay, they produce bacteriocins which have the ability to kill other bacterial cells which lacks the bacteriocin. Okay, so these are the colicin plasmids, and these are generally present in the E. coli and thus named as col plasmids. Then fourth one is the degradative plasmids. Degradative plasmids because these plasmids have the ability to break down different types of chemicals and industrial waste products or industrial substances, for example toluene, camphor, salicylic acid. So whatever types of waste byproducts generated by the bacterial cells during the industrial fermentation, you can say, for example, so degradative plasmids helps in the degradation of these types of substances. Then the last one is the virulence plasmid. That means these plasmids have the ability to produce virulence factors. Some sort of virulence factors are produced by these plasmids, thus can infect other bacteria or plant cell or animal cell or human cells. Okay, so example is TI plasmids, that means Agrobacterium tumefaciens possess TI plasmids inside its bacterial cell, and thus can produce crown gall bladder disease in the plant cell. We know that TI plasmids can cause virulence in plant cell, thus can produce crown gall bladder disease. So these are the types of the plasmids, and the last is the functions. So we have seen the functions in earlier slides already. So let's see in brief again. They can be used as a vectors. They have the ability to produce antibiotic resistance genes inside it, They can degrade industrial chemicals or industrial waste byproducts. They can be used as a gene therapy because they can be easily manipulated. That's why they can be used in the gene therapy, genetic engineering. They can be used in drug delivery because they have the ability to do conjugation, thus can be used in the drug delivery and hormone productions. And they are the very important source of horizontal gene transfer. So this is the structure, function and types of the plasmid. This lecture will explain you about the structure, functions and types of the plasmid. Thanks for the watching.